So
Okay.
our next speaker is Christoph Muller from EMBL Heidelberg, and he's going to talk about structural and functional analysis of RNA polymerase 1 and 3 transcription complexes.
Okay. I would like to thank the organizers for inviting me. It's very nice to be back here. And so I would like to talk a little bit about our work on the RNA polymerase 1 and 3. And as you might all know that in eukaryotes there are 3 different RNA polymerases. There's RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. And I guess you all know RNA polymerase 2 because it makes messenger RNA, which is then translated by the ribosome into proteins. But then in addition, there's also RNA polymerase 1 and RNA polymerase 3. RNA polymerase 1 makes these long transcripts, ribosomal RNA, which then are built into the ribosome. And RNA polymerase 3 makes short transcripts, for example, tRNA, but also 5S RNA and 7SL RNA. And so it makes, which are then required for protein synthesis. And if you wish, pol 1 and pol 3 have almost opposing activities because pol 1 makes these long transcripts, whereas pol 3 makes very short transcripts. And there it's important to produce many copies through a process which is called facilitated reinitiation, where the polymerase stays attached to the pre-initiation complex and very quickly cranks out different copies of tRNA. So just to give you an overview about which are the polymerases doing what in the cell and about the different transcriptional activity, if you look at the number of genes which are transcribed by pol 1, pol 2, and pol 3, it's clear that only a very small, most of the genes are actually transcribed by RNA polymerase 2, and only very few genes are transcribed by pol 1 and pol 3. However, if we look at the transcriptional activity, the situation is very different. So you see 60% of the total transcriptional activity is actually spent by RNA polymerase 1 to make these long transcripts. And there's about 15% spent by RNA polymerase 3. And the rest of the transcriptional activity is spent by RNA polymerase 2. But if you look more closely, half of it is spent to make ribosomal proteins. So in total, there's a lot of transcriptional activity going on in the cell, in the growing yeast cell, mainly to just make the components which are required for protein synthesis. If you go then one step further and you look at the RNA level, so you see most of the RNA which is present in the cell at a given moment has been actually produced by pol 1. So 80% of the total RNA transcripts are made by pol 1, and then they're built into the ribosome. And still these 15% of pol 3, whereas the messenger RNA is synthesized, is translated, but then very quickly gets degraded. So at a given moment, there's relatively little messenger RNA present. So pol 1 and pol 3 also need to be very carefully regulated during cell growth and division. And so there are different tumor suppressors and oncogenes which converge on the pol 1 and pol 3 transcription machinery to keep that keep that controlled. Some of them directly target the polymerases, but in most cases they actually target the pre-initiation complex, and that's one of the motivations to also understand the architecture of the pre-initiation complexes. So RNA polymerases are multi-subunit enzymes, and this just gives you an overview of the subunit composition. Polymerase 2 contains 12 subunits. And so all of them, they have these large subunits which form the core together with a couple of other subunits. Pol 1 and pol 3 contain 14 and 17 subunits. They contain additional subunits because general transcription factors kind of invaded the enzyme and then became permanently attached subunits. And so in the case of pol 2, we have 2 TF2F-like subunits, which are called A49 and A34. In pol 3, we also have the two TF2F-like subunits, C37 and C53. But then in addition, we have a TF2E-like complex, which is formed by 3 other subunits, C31, C82, and C34. And just to give you also an overview or a feeling about the similarity between the different subunits. So these large subunits, they share about 20 to 30% identical amino acids across the pole 1, pole 2, and pole 3. However, there are also subunits which are shared, like some of these small ones, ABC, so they are present in all the 3 RNA polymerases. And then we have some subunits, like the stalk subunits, which have very little sequence homology, which is almost not detectable. So we are studying the yeast enzymes, and we started out as crystallographers, so we always need large amount, and so we're growing kilogram of cell paste, of yeast cell paste in a fermenter. This is a 150-liter fermenter. And we can then biochemically purify these polymerase 1 and 3. And you see here, these are gel filtration profiles where you see all the 12 subunits present in, all the 14 subunits present in pole 1, and all the 17 subunits present in pole 3. And so this material is obviously ready to go for structural analysis. And at first, I would like to talk, say a few words about our work on RNA polymerase 1. RNA polymerase 1, many people know because of the so-called Muller trees. Muller was an electron microscopist, and he discovered these tree-like structures. And they are formed because the pole 1 gene, there are multiple copies of the pole 1 gene, which is arranged in tandem. And so we have tracks of pole 1, which is transcribing here. And these are the transcripts which go out and which form the branches of the Christmas tree. And at the end of the branches, there are RNPs, which introduce further modifications. Some of them Bruno has talked about yesterday in his talk. And so this is, again, the scheme where you see how these Christmas tree-like structures are arranged. So a couple of years ago, we solved the crystal structure of RNA polymerase 1, and this was possible due to a lot of data sets collected here at the ESF synchrotron, but also in other synchrotrons. And just to give you an overview again about the structure, so these are the 2 large subunits here. Here at the bottom is this TF to F-like subunit here, the dimerization part. You see these very long extensions which go out here at the outside of the core enzyme. And here at the top is the stalk. The active center is actually some here. And so here is a DNA binding cleft, and downstream DNA enters here. It makes a 90-degree kink, which you'll see in a minute closer, and comes out towards you. So this RNA polymerase 1 was a first start, and what was surprising when we looked at the structure, that it had a very large DNA binding cleft, that the DNA binding cleft was very widely open. It was so far open that the bridge helix, which is a helical element which bridges the 2 parts, that it was unwound in the center, and that was for the first time seen. It has been proposed, for example, in the bacterial polymerases, that there is some conformational change going on there. They see a kink and some loosening of this helix, but in this case, it was completely unwound. And so one of the interests was then to study to see how does polymerase 1 looks when it's bound to a transcription scaffold. And at the time, we said, OK, let's do this with crystallography. But in parallel, we also started to do cryo-EM. And as nowadays is often the case, cryo-EM was just faster, and we obtained complexes of pole 1 bound to different transcription scaffolds. And we used one scaffold which has a 3080 base pair duplex DNA, and then it has this mismatch region in the center where the DNA are not mismatching. This is an artificial bubble, and we can hybridize RNA to it. And when we could solve the structure, in this case, of the elongation complex at 4 angstrom. And we had another transcription scaffold which was longer. And in this case, we obtained, in fact, 3 different structures. One, an open complex, where we see the DNA hybrid, but we don't see the RNA bound to it. We see an elongation complex, which is very similar as the elongation complex we obtained with the shorter scaffold. And we saw also a minor population of about 8%, where we had an additional domain. this tandem winged helix domain, which is at the C-terminal part of the C-terminal end of one of these TF2F-like factors. And this structure had been, this domain, there was a crystal structure known from this domain, but in our crystal structure of the entire pole 1, this was a flexible part, and for reasons which we still don't understand, we found now here a population where it's bound to the DNA and where we can see it in greater detail. So this gives you a view on the open complex. So you very nicely see the density for the downstream duplex, and you see, but then the transcription bubble forms, so the double strand unwinds, and we don't see any density for the upstream DNA. But in the other reconstruction, which used actually a shorter DNA duplex, but nevertheless here we see the downstream DNA as well, but we also see the upstream duplex. And here we see now very nicely how the RNA is synthesized with the 3' end here in this position. We can compare these conformations now with the structure which we initially obtained by X-ray crystallography. And what we see there is that the DNA binding cleft is gradually closing. So here we have a 42 angstrom distance between the outer part, and then in the open complex, the DNA cleft closes, and it further closes in the elongation complex when it's then in the process of transcribing the DNA duplex. So this, if the movie is working, yes, you see now, this is again shown in a very simplified movie, but you see how the conformation changes happen. This is the open conformation, the crystal structure, and once it's elongating, kind of converge together. The two half sides come together and enclose the DNA. What is also happening in this process is that the bridge helix, which we said at the beginning is unwound, it gradually forms a helix. So at the beginning it's unwound, and then in the transition from an open complex to the full elongation complex, this bridge helix completely folds. And what we also observe is that, and there I have to extend that first a bit, or explain that first, the RNA polymerase 1 contains one subunit which is called A12, and the C-terminal part of A12 has an intrinsic RNA activity. And this is important because if the pole 1 kind of backtracks, RNA needs to be cleaved. And in the case of RNA polymerase 2, there's an outside factor binding in this case, which is called TF2S, which binds into the active center. Whereas in pole 1, this RNase activity is a built-in feature of the RNA polymerase 1. And so we have the C-terminal domain, which binds in the active center. And what we see is that in the open complex, when the DNA is half closed, the DNA binding cleft is half closed, the C-terminal domain still sits in the active center. And it's also, we can also see that in the apoform. But then if the elongation complex completely forms, then the C-terminal domain gets expelled and is no longer present in the active center. And so we could observe that here. And finally, I'll say a few words about this tandem winged helix domain. So this was, as I said, only a fraction of the particles actually contained this conformation. And it's in a conformation where the DNA binding cleft is completely closed, and we think the DNA binding cleft has to close so that the linker here finds its very nice place, and we can then position the tandem winged helix here, interacting with the upstream DNA and forming interaction here with the upstream DNA. And it is still, we still think this is an intermediate situation. Generally, the tandem winged helix domain has been described being important for promoter recognition. And so, and we still don't know why we see this in some cases, but not in all of them. Okay, now to move on. So the pole 1 was we thought, a very nice result by itself, but it also gave a very nice model for RNA polymerase 3. And so we solved in 2010 a 10 angstrom cryo-EM structure of RNA polymerase III, and so we could now take the pole 1 crystal structure and fit it into the density of pole 3. And if we do that, you see that all fits very nicely. So the TF2F-like part, for example, sits there very nicely. However, there is one part missing, and this is obviously the TF2E-like complexes which are present in RNA polymerase III in addition to these 3 complexes. Fortunately, at the time, there was a crystal structure solved by Sebastian Fribourg in Bordeaux of the human homologue of C82. And so from there, we knew that it contains 4 winged helix domains and a coiled domain. We knew through secondary prediction that C34 contains 3 winged helix domains, whereas C31 is mainly disordered. However, the way how these 3 complexes are arranged in this density wasn't clear, and the density was also not good enough to answer these questions. So we moved on and solved the structure of RNA polymerase III in 2 forms, in an elongating form bound to a transcription scaffold, and also in its apo form. And this shows you the FSC curves for both structures, and also the local resolution which had mapped onto the structure. And what you can see very clearly is that whereas the core is very well defined and we see very nice features here, the outer parts are much more flexible. You also see that the apo structure is more flexible in general, so it seems binding of the transcription scaffold stabilizes the enzyme so that it binds more tightly. But it's clear in both structures these outer parts are extremely flexible, and I think this is one of the reasons why we were never able to crystallize the pole 3 enzyme, and I can tell you we really tried very, very hard to do that. This shows you the electron density. So again, you have seen similar pictures from other people. So you see in the center, the density is very nice, and you have really sidechain resolution, and you can build very precise models. However, the outside parts, the density for the outside parts are much more flexible, so there's more mobility there. So the resolution is not completely uniform, as it is often the case in these very large assemblies. So this shows you the overall structure. So again, you see the 2 large subunits here, and you see the downstream DNA here, the sharp right-angled kink here with the DNA duplex, the upstream DNA coming towards you, and the RNA which is synthesized here. This is the TF2F-like factor, which you now recognize, and this is now we see TF2E, these TF2E-like 3 subunits, how they are built, where they are located here, with the 4 winged helix which are present in C82. For C34, we see only 1 of the winged helix, the C-terminal winged helix, whereas the 2 N-terminal winged helices are disordered, and we'll come back to that in a minute. And so now we have the full gallery of the eukaryotic RNA polymerases, pole 1, pole 2, and pole 3. And we can again ask the question, so what makes pole 1, pole 2, and pole 3 particularly suitable for their respective tasks? And one of the, and obviously there's a lot one can say there, but I just want to highlight a few aspects. One thing we noticed that the downstream DNA in RNA polymerase III is extremely tightly bound and is very well ordered. And the reason for that is, or it is entered by different regions of the RNA polymerase III. So from this clamp and jaw from the large subunits, but then importantly also extensions from these DF2E-like factors, these winged helix domains, they reach down and they enclose the DNA duplex, and we think this is functionally important. This might contribute so that the polymerase can stick to a DNA substrate and not dissociate and has to reinitiate, and so explain or contributing to this process, what I said at the beginning of facilitated reinitiation, where pole 3 stays attached to its transcription scaffold. This again shows you the TF2E-like factors, and so you see in total we have 8 winged helix domains, and it's these TF2E-like factors which is important for, plays a very important role in opening the DNA duplex so that transcription can occur. I should also say for the third subunit, C31, we can only place we can only put sequence in a relatively short stretch here in this helical part here. And here we see additional density, but diffuse density between the stalk here and the TF2E trimer. But it's rather flexible. So this we are still not able to locate all the to put sequence in all these areas, and it seems to function as a clue between these two elements. So the next step now is to look at pole 1 and pole 3 pre-initiation complexes. So pole 1 and pole 3 both use their very distinct sets of general transcription factors to recruit the polymerases. And in the case of pole 1, it's the upstream activating factor, which then recruits TBP, core factor, and then through a bridging factor RN3, RNA polymerase 1. And this core factor contains a TF2B-like factor, which is called RN7, beta propeller containing protein RN6, and a TPR protein RN11. And so we were, in a first step, assembling this kind of minimal pre-initiation complex comprising pole 1, the bridging factor, and core factor. And this is shown in the next slide, so we can make such a complex. These are first negative stain images, and then we obtained a cryo-EM reconstruction of this pole 1 pre-initiation complex. The overall resolution which we obtained is at 4.4 angstrom resolution. But then we can make a focused refinement, and so we obtain a 3.85 resolution structure for the pole 1 and RN3, and about 4 angstrom for the core factor. But if you map the local resolution onto the core factor, it is clear core factor is much more flexible than the pole 1 core, which turned out to be, slowed us down in a certain sense. And this shows you the overall structure of this pole 1 pre-initiation complex. The big surprise was that the TF2B-like factor binds much further upstream, as it is the case in RNA polymerase 2. So it is really distance. In the case of pole 2, it packs directly against the pole 2 enzyme, whereas in this case, it is actually distant from the site. And RN11, this TPR protein, seem to function as a spacer. What is also clear, what you can also see, is that the DNA doesn't have a bend, and this is shown here again. So as you see again the TF2B-like factor with the T2 cyclin domains binding here, quite in the distance with the R11 function as a spacer. If you compare the course of the DNA between the pole 1 peak and the pole 2 open complex, as it's observed in a pole 2 pre-initiation complex, you see that in pole 2, the DNA runs much more further outside. And it does so because TF2B is bound in this position. So it really runs outside and has to go quite some way to get into the active site. Whereas in pole 1, in the pole 1 peak, TF2B sits here further outside, and the DNA runs in a position which is actually very similar to the DNA conformation which we see in the elongation complex. Again, we think this is also might be functionally relevant because pole 1 has to be very quickly ready to go, and so in the pole 1 pre-initiation complex, it is the DNA is already positioned to be ready to go immediately in the active center. But it's a situation which has been really unexpected and very different from the pole 2 pre-initiation complex, which we saw before. While we're thinking about this and we're just finishing the paper, as another structure got published from the Kramer lab in Cell, and now recently there has been yet another structure. And our manuscript is coming out soon again. If we superimpose all these structures, the structures look very similar. However, there's a lesson there, and one of the lessons is that the Kramer lab, in addition, were able to crystallize the core factor, and that obviously helped them a lot when they interpreted the electron density whereas we had a relatively roughly 4 angstrom resolution electron microscopy density, and it was just difficult to put sequence in all the parts. In some parts, we could do that very nicely through homology, but just one example, for example, we had these helices there, and they are linked through a flexible linker, and it was just not possible for us to assign definite sequence there. And so crystallography is still a very important technique, and I think one has to be careful to keep both techniques at a high level if one works on these large complexes, which sometimes is a challenge. So this summarizes again the situation in pole 1 and pole 2. So you see in pole 2 we have a strong kink here, which is introduced by the TBP binding here. In case of RN7, in the case of pole 1, the TF2B-like factor binds much more upstream, where in the case of pole 2, it directly packs against the enzyme. And so the architecture is very different. As a next step, we are now looking at the pole 3 pre-initiation complex. In the pole 3 pre-initiation complex, as it's found in type 2 promoters, those are promoters which are important for transcription of tRNA. It is TF3C which positions TF3B, and TF3B then recruits RNA polymerase 3, and TF3B has an extremely tight binding binding to DNA if it's in complex with RNA polymerase 3. And this just comes back to what I said at the beginning. So many of these tumor suppressors and so on, they actually target the pole 3 pre-initiation complex, so understanding the architecture there is important and is also of medical relevance. And so we obtained a cryo-EM reconstruction now at 3.8 angstrom resolution of the minimal pole 3 complex, so comprising pole 3 and TF3B. And so this is the overall view, and so we have BRF1. So I should say TF3B contains 3 different proteins. BRF1, which is TF2B-related factor, BTP1, which has a large disordered N-terminal part, and then a more ordered C-terminal part, and pole 3, different to pole 1, also contains TBP. And so we can assemble this complex, and if we did see that, so this is the RNA polymerase 3, this is TBP binding here, with introducing a sharp kink here in the DNA, and then the TF2B-like factor BRF1 binds in the top, and then as well as BTP1. One of the surprises of the structure is that the 2 C-terminal winged helix domain of C34, which were disordered in the pole 3 structure, suddenly become ordered, and they kept in place through these coil domains of BTP1, and so they fix them like chopsticks, fix them here at the body. And another aspect which is really surprising in this structure is how closely the DNA is interacting with TF3B. So again, these are the cyclin domains here. On one side, you have TBP on the top side, and BRF1 covers it from these 2 sides. So the DNA is really by 360 degrees entered by this factor, and that's why people already was suspecting something like this. It's like a proteinaceous cage around the DNA formed by these transcription factors. There are many more things one could say about, but just one aspect I mentioned before, these TF2E, TF2F-like factors, and in fact we can see now this is again this is now a comparison between the pole 3 pre-initiation complex and pole 2, and so you see the course of the DNA is actually quite similar, different from the course in pole 1. And C82 really plays a TF2E-like role here, contacting the DNA through a loop here, similar as TF2E does it. However, for the C-terminal domain of TF2F contacts here the DNA, and in pole 3, the C-terminus doesn't contain such a winged helix, through a winged helix domain, doesn't contain such a winged helix domain. Instead, these the N-terminal domains of C3040, which are also winged helix domain, those contact the DNA substrate. And so there are things which are similar, but things are also definitely different in the two complexes. And this just summarizes again now the three polymerase pre-initiation complexes as we have now determined. So there is the pole 1 peak with this quite particular situation with the TF2B binding more at the outside. And then we have the situation if in pole 2 with TBP and TF2E and TF2F contacting from both sides. And finally, the situation in the pole 3 peak where the C82 is the TF2E and C34 contacts the DNA like TF2F in the polymerase 2. And with that, I would like to thank the people who did the work. First of all, I should highlight Carsten, who will also give a talk now. Carsten and Arjen and Wim, those people helped us a lot in getting into cryo-electron microscopy and were absolutely critical to solve for us as a crystallographer moving into the cryo-EM field. This is Niklas, who solved the pole 3 structure, and Lukas did the transcription complex with pole 1, and Yasha the entire pre-initiation complex, and Matthias the pole 3 pre-initiation complex. And with that, I would like to thank you for your attention.
Thanks, Christoph, for the great talk. Are there questions, Andres?
Here, Christopher. Here.
Over there.
Here.
Sorry.
Very nice talk. Transfer RNAs are one of the product of polymerases. So by looking now at the gallery of pole 1, pole 2, and pole 3, what can you say about the preference for one of these polymerases for making this product RNA, which has a very well-defined structure?
Well, I mean, I think the main difference is really on the level of the promoter. So the questions of the promoter recruitment and how the different ways the promoters assemble. So our conclusion from our work here is that pole 1 and pole 3 are much more ready-to-go enzymes in the sense that they have their general transcription factors built in. They can assemble their pre-initiation complexes very quickly, and I think this is important to be responsive and to be able to produce very large amounts of these short transcripts. We looked at if there are, for example, tRNA stabilization sites and so on the RNA polymerase 3, and we have some indications there, but nothing so conclusive. I mean, there were initial thoughts that the pole 3 tRNAs would bind towards the stalk and on the shoulder of the polymerase 3, and we tried to make complexes with tRNA there. That so far hasn't been successful.
Question here. So would you call C82 a helicase? I mean, you say that it opens the DNA.
It's not an ATP-dependent helicase. It is a. I mean, it's the combination of multiple interactions which stabilize the DNA duplex in such a way that then it can the DNA in such a way that can unfold and that single-stranded DNA templates are formed. It's not an ATP-dependent helicase in this sense. No, it's not a helicase.
Do you see any difference on the DNA level, like the transcription bubbles are the same, or I mean, like the promoters must be different for ribosomal RNAs and the tRNAs and the mRNAs. Do you see any difference in the complexes on the bubbles? And
I mean, one problem or one limitation in many of these transcription scaffolds which we provide, we kind of preform the transcription bubble. So the transcription bubble is formed through these mismatching DNA pairs, and so in this way you preform the DNA bubble. In both pol1 and pol3, we can actually provide closed, and that's also to the previous question, we can provide closed DNA duplexes, and the polymerase by itself is able to open these complexes. And so so in this case, often we have difficult to see nicely the transcription bubble. We think because they are bubbles of varying size, they are not completely uniform in the size, in the transcription bubble size, which is then formed.
Yeah, last question.
Yeah, and Chris, I was wondering about the backtracking that you alluded to, the 812 subunit on the pol1. So is that a common feature of all polymerases, or is there any factor that would try and avoid backtracking? Or is that, I mean it's a naive question, but just to understand the mechanism.
I mean, pol1 and pol3 both have these, essentially have the C-terminal domain of TF2S. This is the pol2 outside factor fused to the C-terminus of a subunit A12, C12, C11 is called in RNA polymerase 3. So they both have this built in, whereas in pol2 it comes from an outside factor.
Okay, and there's nothing that blocks that, or in a systematic way?
No, I think this is, I mean, it is blocked. It's obviously blocked if the pol1 is elongating, because then the DNA binding cleft is blocked and access is prevented. So the idea is that once you have backtracking, it must partially open, not that much, partially, so as we see it in an open complex, and then suddenly you get access to the active center and you can function there. So it's the conformation of the polymerase essentially which